Macromolecules are very large molecules important to living organisms. Most macromolecules are built by joining smaller molecule subunits, which are often called monomers. The term monomer basically means single unit. There are four major classes of macromolecules that are important in biology, and they are the carbohydrates, the lipids, proteins, and nucleic acids. The rest of this slideshow will look at each of these macromolecules individually. Carbohydrates are often used by organisms for energy storage or for cell structure. They come in different sizes. Small carbohydrates are often called monosaccharides. The term monosaccharide basically means single sugar. Long chains of monosaccharides can be formed to build a polysaccharide. Polysaccharide basically means many sugars. All carbohydrates consist mostly of three atoms: carbon, hydrogen, and oxygen. As an example, let's look at the structure of a commonly used monosaccharide called glucose, which consists of 6 carbons, 12 hydrogens, and 6 oxygens. In reality, many monosaccharides like glucose form rings. For example, the glucose ring structure consists of carbons 1 through 5 that are part of the ring, with carbon number 6 that is actually not part of the ring. The 6th member of the ring is one of the oxygen atoms in glucose. To form a polysaccharide, multiple monosaccharides are joined together and are linked together by covalent bonds. The chemical reaction that is done to form any covalent bond is called a condensation or dehydration reaction, so named because a water molecule is removed in the process of forming the bond. For example, to link two glucose molecules together, the first carbon of a glucose is linked together to the 4th carbon of the second glucose by a condensation or dehydration reaction. The next few slides will review some commonly found polysaccharides in biology. The first two are used by organisms as a form of energy storage. Plant cells form starches by linking up thousands of glucose molecules. Starches can be branched or unbranched depending upon how these glucose molecules are linked together. Animal cells, such as your liver and muscle cells, will form a highly branched polysaccharide by linking many glucose molecules together, similar to a way a starch is built. This large polysaccharide is called glycogen. The next two polysaccharides are used for structure. When glucose in a slightly different configuration, called the beta configuration, is linked together in long chains, this forms a plant material called cellulose. Cellulose is the main component of all plant cell walls. Some organisms make long chains of a modified version of glucose to form a material called chitin. Chitin is a polysaccharide that is used to build the exoskeletons of animals such as insects, spiders, crabs, and shrimp. It is also found in the cell walls of fungi. Lipids are macromolecules that are extremely diverse in their structure. Overall, most lipids are hydrophobic, meaning that they do not mix well with water. Although there are many different types of lipids, this slideshow will only focus on 3 of them: the fats, phospholipids, and steroids. Fats can also be called triacylglycerols or triglyceride. They share a common structure, consisting of one glycerol molecule, which has 3 carbons, and 3 long carbon chains, each called a fatty acid. There are 2 basic types of fatty acids: long carbon chains that lack double covalent bonds, called saturated fatty acids, and ones that contain at least one double covalent bond, called unsaturated fatty acids. Saturated fats are ones that contain saturated fatty acids. The presence of saturated fatty acids causes the overall shape of the molecule to be very straight, kind of like the capital letter E. This very straight structure allows saturated fats to pack tightly together at room temperature. so they stay solid at room temperature. The presence of a double covalent bond in an unsaturated fatty acid can cause kinks in the shape of the molecule, such as in a cis unsaturated fatty acid. Fats that contain at least one cis unsaturated fatty acid will not be able to pack as tightly together, and thus will stay liquid at room temperature. Atoms can be arranged a little bit differently around a double covalent bond to make the kinks a little bit straighter in the fatty acid. These fatty acids will behave a little bit more like saturated ones. The three fatty acids in a fat molecule do not have to be exactly the same. Each fatty acid is held to glycerol by a covalent bond, which was formed by a condensation or dehydration reaction. Phospholipids have structures similar to fats. They consist of one glycerol molecule with two fatty acids attached. In place of a third fatty acid is a group called a phosphate group, which has a negative charge. The presence of this negatively charged phosphate group makes a phospholipid partially hydrophilic or water-loving. All cell membranes consist of phospholipids. The phospholipids in a cell membrane are arranged in two layers, which is sometimes called a bilayer. The phospholipids in one of these two layers of a bilayer are arranged so that their hydrophilic heads are together and their hydrophilic tails, the fatty acids, are together as well. The bilayer is arranged so that the hydrophilic heads of one layer is pointing away from the hydrophilic heads of the second layer. Steroids are a third major type of lipid. All steroids share a common chemical structure, which consists of 4 interconnected carbon-containing rings. The steroid cholesterol is a major constituent of cell membranes and is present to maintain the membrane's flexibility. Steroid hormones, such as estrogen and testosterone, act as cell signals. Proteins are macromolecules that are essentially long chains of monomers called amino acids. Different proteins do different jobs in organisms, including acting as enzymes, hormones, channels, or receptors. There are 20 possible amino acids that can be used to build a protein. They all share a common structure. Each amino acid has a central carbon atom that is attached to an amino group on one side, a carboxyl group on another side, a hydrogen, and a last group called an R group. The R groups are groups of atoms that make the 20 amino acids slightly different from each other. To form a protein, amino acids are joined together by covalent bonds. Again, these covalent bonds are formed by condensation or dehydration reactions. The covalent bond that joins two amino acids together is sometimes called a peptide bond. Thus, a long chain of amino acids, or basically a protein, sometimes called a polypeptide. In order for a protein to function properly, it can't be left as a polypeptide. It must be folded into a distinct three-dimensional shape. There are different levels of bonding that all participate to form the distinct three-dimensional shape of any protein. These are called the levels of protein structure. We've already covered the first one, or the primary structure of a protein, which is basically amino acids joined together by peptide bonds. The secondary structure of a protein is where this polypeptide chain is folded even further into one of two different shapes. One type of secondary structure is called an alpha helix and looks like a spiral. The second type of secondary structure is called a pleated or beta pleated sheet and looks like accordion pleats in a skirt. Both of these secondary structures are held together by hydrogen bonds. When a protein is folded even further than its secondary structure, this is the tertiary or third level of protein structure. Many different kinds of chemical bonds can hold together the tertiary structure of a protein, including covalent, ionic, and hydrogen ones. In organisms, many proteins do not work alone. When multiple proteins associate together, this forms the quaternary structure of a protein. Hemoglobin is an example of a protein quaternary structure. Hemoglobin consists of 4 globin proteins associated together and working together as a single unit. Nucleic acids act as a form of information storage. They can be passed on from one cell to another and from one organism to another. There are 2 major types of nucleic acids. One is called DNA and the other is called RNA. Both DNA and RNA consist of long chains of monomers called nucleotides. DNA and RNA nucleotides consist of 3 major parts. The first part is a monosaccharide. In DNA nucleotides, the name of the monosaccharide is deoxyribose. In RNA ones, the structure of the monosaccharide is slightly different and is called ribose. Attached to one side of a monosaccharide in any nucleotide is at least one phosphate group. Attached to the other side of the monosaccharide is a nitrogen-containing ring structure called a nitrogenous base. Pyrimidines are nitrogenous bases that are single rings. In DNA, there are 2 possible pyrimidines, cytosine and thymine. In RNA nucleotides, the 2 possible pyrimidines are cytosine and uracil. Purines are nitrogenous bases that are double ring structures. Adenine and guanine are the 2 possible purines in both DNA and RNA. Nitrogenous bases are able to form hydrogen bonds with each other in a specific way. For example, adenine will form hydrogen bonds with thymine, and guanine forms hydrogen bonds with cytosine in DNA molecules. This is especially important to form the 3-dimensional structure of DNA. In RNA, the nitrogenous base adenine forms hydrogen bonds with uracil. To form a nucleic acid, nucleotides are joined together by covalent bonds. A covalent bond is formed between the monosaccharide of one nucleotide and the phosphate group of the next one. The resulting nucleotide strand has a head, which is sometimes called the 5' end, and a tail, which is sometimes called the 3' end. DNA consists of 2 strings of nucleotides, while RNA is single-stranded. In a DNA molecule, the 2 nucleotide strands are held together by hydrogen bonds between opposite nitrogenous bases. Notice the orientation of the 2 strands relative to each other, that their 5' and 3' ends are in opposite directions. This is sometimes called an anti-parallel arrangement. In prokaryotic cells, like bacteria, the DNA is in the cytosol and is not housed in a membrane. In eukaryotic cells, such as human ones, much of the DNA in the cell is housed in a structure called the nucleus. DNA functions as stored information in a cell. Much of the information that's stored are protein recipes. Each and every recipe encoded in the DNA is called a gene. DNA is not only stored information, it is inherited information. It can be passed from one cell to another and from one organism to another. RNA molecules are generally smaller than DNA ones. They are also single-stranded. Instead of 2 strands of nucleotides, they consist of only 1 strand of RNA nucleotides. This RNA nucleotide strand has a 5' end and a 3' end. And because of hydrogen bonding between nitrogenous bases, the strand can be folded into a distinct 3-dimensional shape.